Okay. Welcome, everybody. Good morning. I hope that you have your coffee. You will need it. And I will have, I'm glad I have the opportunity to tell you everything that I know so far or what I am learning about Eriophyid mites. Definitely it's a learning road, and it's getting complicated because these creatures are well adapted to do what they do. Eriophytes are very small. They are in all kinds of shapes and sizes and colors. They spend all the time feeding and around the plant. There is some complex in modification of the mouth part. There are the pulps became part of the natosoma, very attached to them, have a lot of structures in the way that they pressure and feed. However, the stylus are not too long. That usually, often the more complicated ones are only 5 microns to 10 microns. The more vagrant are longer in the size. All are phytophagous. It's a very old group. We didn't know that. We were thinking that there was a very advanced group, but no, there is very old. They are almost evolved at the same time that the plants were evolving. That is a concern because they are well adapted to be in that way. They are not the spider mites. They are not in that group. However, there are plant feeding, all of them. And IDEs were considered close to them. However, more and more we are finding that it's not. At the same time, we are trying to understand and use molecular techniques to figure out where is the association, where is come from, how they evolved, how we're able to be so successful, and has been complicated because, as I said, it's a very old group. It's showing up some apomorphics, synapomorphics, and some areas that we still not clear how to separate them, but definitely it's a group well adapted to the association with the plants. In general, there are 100 to 200 microns in length, and the width of the body is just 50 microns. That give us a lot of headache of try to confine them and work with them or get molecular data from so tiny little mite. There are smaller than other tarsonemids that you know, like the broad mite, Polyphagotarsonemon latus. The broad mite is almost twice the size of the Eriophyid, and usually they are not visible for us. Numbers, my gosh, in numbers, a couple of years, in 1994, there were said 300 genera and 3,000 species. Today, we know that there are close to 6,000 species, almost describe it, 6,000 species probably describe it. And the other problem that we had is if you multiply the number of species of plants and you guess that there is 1 to 3 species of Eriophyid mites specialized per host plant, you are dealing with close to 1 million species of mites. That is something that helps us to understand what is going on. How we study them, how we describe them, that is part of the problem. It's very cosmetic. The lines over the dorsum, the lines over the prodorsal shield that you can see here. This is the prodorsal shield. In our case, our Phylocoptes fructifilus, look how reticular is the body with another ones that are more linear. But that is what the things that we need to look. And unfortunately, sometimes the variation of some of these reticles, they indicated that it's a different species. We need to be more aware of other characters like the annuli, the segments, and the length of the front of the shield, like this one, if it's pointed, if it's rounded. The leg is very reduced, only 2 pairs of legs. However, there is a case that we know that there is one Eriophyid mite around that somebody captured and took pictures with 4 pairs of legs, but unfortunately still disappeared. It's associated with poison ivy. If you collect a Eriophyid poison ivy, let me know. The genitalia. Genitalia is very important. All the top ones are females, all the different kinds of genitalia of different species, and the genitalia of the males, all the lower ones, this one, and this one, males too. And the color, the shape, the form, the segmentation, the lines over there help us to identify them. Spermatophores, it have a geometrical form, octagonal, pentagonal, all that also help us to identify species. Unfortunately, you need an electronic microscope to really see this kind of detail. That complicate our study. However, look where are the spermatophores located around the stomatas and around areas of the plant that are very important for us to know how they might use the plant to protect themselves and reproduce. Types, we have several types. We can have a small annuli. This is the annuli. Annuli is the segmentation way that they're tiny, little, not too much spread, or can be very heavy, thick, that indicate to us these two that are exposed, that they can survive outside, when these two are more for galls and buds and flowers like the ones that we are working. Other problem that we have is the cycle, how the cycle is, how the weather and the 4 seasons interact with these mites. Phylocoproctus fructifilus is adapted to 4 seasons. It's a mite of 4 seasons. It's not a mite of just 2 seasons. That is one very interesting point here that perhaps is affecting the mite to spread more south. It needs the fall, and in the fall it will come the different forms for resist and tolerate the winter. In this case, for example, here we have Aseria Antocoptes that is associated with Cirsium, and you can see the 2 forms really clear, the winter form and the summer form. You can see that all that is well programmed in base of the phenology of the plant. They know what they are doing and they know where they want to go. Shapes, like I said, as with millions of species, we will have many kind of shapes, many kind of forms. Apparently, something that we notice is that the most simple forms, the more rounded forms, are the ones associated with viruses, when the other ones that are more exposed are not associated with virus. But these need to be corroborated. That is part of the learning that we are. Here, our 4 more studied mites that we know, and Antocoptes here on the left, Phylocoptes fructifilus, Calacaros and Vitacus. These 4 genera are very important in plant feeding, and some of them have been associated with viruses. Host specific, that is one of the things that we know. These mites, definitely there are some of them are host specific, especially the ones that are associated with viruses and with galls and eriniums and things like that are the host specific. The non-host specific are vagrant, do not do that problem. The response of the feeding, we still not know if it's response for the plant, if it's a toxin from the mite, if it is a symbiont from the mite, if it's an association between virus, symbiont, and mite feeding, toxins, change of auxins, cytokinins, all that is part of what you are doing. That's part of what you are studying. The salivary glands, Eriophyid mites have 3 salivary glands. That means that we now know also where the virus go or where the symbionts are located with respect to that. And feeding, you can see the holes here in this leaf, all the different holes where the mite has been feeding, and the spermatophore around. That means that they have been very busy. This looks a loss of time in consumption. Perhaps we can talk about that later with questions, because this is the one of Phylocoptes that have the virus. If they have the virus, maybe he need to speed more to be able to clean the virus. Symptoms, I will show you a couple of symptoms just to get an idea. These are the eriniums. This is erinium by Aseria. This is erinium by Culops with galls. Also at the same time, inside the galls, there is erinium. These are erinium by another Seria on ribes. And here is what we know. Here is what I want to call your attention here. Here in the left, in color, is our mite attacking rows. Look the erinium, look the trichomes, look the form of the trichomes, that erinium is also the trichomes. Look the glandular trichomes, compare that with Eucalyptus simplex in Ruellia, compare that with the beech tree also feeding here, and this, believe it or not, this is glandular trichomes too. And on the right, you can see oleander, erinium also, and compare that. Why I am pointing this? Because this is one of the mites that we are studying at the same time that we are studying rose rosette. We are studying the lychee mite. that cause this amazing damage. Believe me or not, all the Erinium on the right, that nice trichons pointed with spicules over it, like you can see this on these guys on the right, that is the normal trichon that is supposed to be the true trichon of the plant. Look, all the on the left is completely deformed. We call deflated trichomes, and all that deflated trichomes is where the mite is located, is where the mite is feeding, not in the other trichome. We prefer this one over the other one. And look that there is one healthy trichome in the middle of the deflated. That really opened the eye for us of what is going on, what is the association, if the trichomes have something to do definitely with the feeding and behavior of the mite. With that, try to also to understand what is the interaction with the predators. Predators, we have the Phattosciurus persimilis that many of you are aware. We have the Galindromus occidentalis and the Neosciurus cucumeris that you can buy, you can use. Unfortunately, all these 3 mites are not good killing the Eriophyid mites. They cannot reach them because they are protected. However, Tideidae, this one nice orange, and excuse me, and here Loria formosa that now is called Brachiotedeus formosus. Sorry about that. Acarology is still learning with the names. And these two indeed are the good ones that they can go and follow the Eriophytes either to the host areas and close areas and can reach them and kill them. Very interesting behavior. And one recent one discovered in Florida on lychee is this Phattosciurus cohenic that is a smaller phytocidal mite that is somehow able to walk over the Eriniums or trichomes and find and pull out Eriophytes. Why is all this important? Because this is what we are learning now with the list of all the plants that have Erinium, all the plants that have the viruses, the virus associated with the mites. Problem. The way that people have been dividing the different species of Eriophytes and the way that they separated the genus is not based on phylogenetics, it's not based on cladistics. It's more like, oh, this one have this leg, or must be a new genus, this one have that thing. And that is part of the problem. That is the reason I try to focus more on the damage and the interaction with the damage than really try to understand what this genus means. With that, I finish. I hope that you have a better idea what we are dealing, how many mites are, and what is the problem. Today, we know that we are dealing with 1 million species of Eriophytes, and the damage that these Eriophyid mites are doing on the roses is very similar to a large number of plants, and the association and the effect on how they are working is almost identical, it's a repetition, and perhaps understanding rose situation will open the doors to understand so many other problems. Thank you.
Thank you very much, Ron. Now you mentioned you found a predator mite that could get the Eriophytes through the trichomes.
Yes,
that was on leech. But you haven't found this on rose or anything similar on rose yet?
Yes. Yeah, we found in roses. And is it another reason I pointed the Tideus. Tideus, one of the surprises that we have is that we get the flower buds, and when we cut them, we were surprised. It was a nice surprise to see inside the bud that is already sealed a couple or 3 or 4 Tideides walking inside, looking for the Eriophytes. That means that indeed Tideus have that ability. They know how also to crawl in. It means that perhaps having roses with less dense trichomes on the flowers, perhaps we will help more the predators to kill the Eriophytes.
Ron, wasn't that inside the rose hip rather than a flower bud?
Yeah, it also was inside the rose hip too also. Yeah. Yeah, the heat was completely dry outside. I cut it, and there were 2 nice, living, happy Tideides inside. A couple of times, sure. You keep cutting, and get the surprises.
Hey, Ron, I got a question for you.
Yes, Mark.
With roses, when we look on healthy roses, we can always find a rose mite, Phalacopsis fructiphilus, but they're in very low populations. We never see any damage, like the damage that you were talking about. On disease roses, especially some of the rosa species, the mite populations, they just spike. They go really, really high. And on some of the rosa species, we get very bizarre rosette symptoms. We just attributed that to it's a different type of rose, and so it's got a little bit of weird symptoms. Do you know if there are any symptoms on roses that are attributed just to the mite if the populations are high? But I don't know how you would get them high without the virus.
Okay. I have experienced a similar mite, that we were able to take out all the predators. There was no virus, only the mite and the host with no virus. And in that ones, when the population really get high, sometimes you will see what we call browsing colors. And browsing is one of the symptoms sometimes that happen when it's only feeding and no predators and no virus.
Well,
and high populations.
That is interesting that you said bronzing, because we have a phenomenon in roses when it is very hot, and that's when Phalacopsis would be high, and it's called blackleaf. We don't understand blackleaf at all, but it starts as a bronzing of the leaves, and we've never been able to attribute to anything, but it seems to be associated with heat and certain cultivars of roses. And so I guess now when there's bronzing, I'm going to ask someone to look at mite populations. Maybe that is what is causing this. Thank you.
You're welcome.
Mark, there's another suggestion that I can make. In order to knock down the populations of the predatory mites, you could try caging a healthy rose with a low population of mites and putting ladybugs in the cage, because they will take care of the predatory mites, but not.
Hear that, Nard?
They won't.
It'll be Nard doing it, not me, but he's listening.
Yeah, but
that's a good idea. So put ladybugs on, get rid of the predators.
Yeah.
Yes.
So I will maybe try that at Beltsville too.
Yeah, I have a question for you. How can we rear the Phalacopsis mite?
Yeah.
Do you rear them in a?
Yeah, definitely. John is doing it in containers, but you also can use a growing chambers, and you can put your roses inside. You can put all your Eriophytes with it. Make sure that you put the ladybugs, that the ladybugs keeping eating the big predators, the phytocidal, and anything that will affect the Eriophytes, and eventually the Eriophyte population that will not have anything to affect them will explode. And we saw that with other Eriophytes in the same conditions in tomatoes with Aculopsis lycopersicii, and with Azaria anthocoptes in Cirsium. We saw also that same behavior, and it works. Eriophytes are very, very fast, very quick, and grow in a really nice way. They know the plan.
Okay, we while I work with the wheat called mite, Escherichia toticella, so we rear them in a cage. They are bigger than Phalacopsis, so that was easier. We transfer the single egg from leaf to the other plants, and rearing the virus-free mites that way. So is it possible for this Phalacopsis also?
Okay, that is fantastic that you have the knowledge of the Azaria toshikella, because Azaria toshikella will use the group of the leaf to put the eggs and grow and move inside the groups. And what they do is that the group close, and when they close, they protect themselves from the predators. However, what you need to look also is, and it's something that I didn't see in that kind of research, is where the mite goes when they start to produce the the grain, because my guess is that when the grain start to come out, I bet you that Eriophyid mice walk into the grain and get inside the grain, but it's something that nobody has been looking at. But that is what I am guessing.
There is a published paper last year showing that mites do get inside the grain and actually survive there over winter.
All right. Okay, perfect. You see, the Sanamagans are doing the same to our roses. They are doing the same.
All right, thank you.
Thank you, John.
Any other questions? Okay, I have one question.
Yes, sir.
What do we know about the other Phyllocoptes species that has been reported to transmit rose rosette virus?
Yes, here we have a little problem, you know, splitter and lumpers and splitters. That mite is so close, so identical to Phyllocoptes fructifilus that could be a haploid species. Okay, it have enough characters to separate them, but now it's kind of localized and is in the western states area. We will know more about it if this one start to display the most common one. But if not, it's a haploid. For me, it's still a very close cousin of the one that we have. And whatever we learn with this one can be used against the other one. That is my feeling. That is the reason I say I am more focused in trying to understand how the virus and the mite interacts and what is the benefit of one and the other one. And here, another thing that was interesting for us is that the role of the trichomes by the roses in the leaf sepals, in the sepals and the flowers may have a strong role of be successful for the Eriophyid mite to pass the virus. That is something that somebody maybe can look at it, because maybe that is kind of mechanical resistance that the plant does to allow more predators to follow the mite and stop that interaction. It's a very interesting situation, no?
Yeah, Oscar.
What we know about the genetic diversity of
What do we know about the genetic diversity of Phyllocoptes fruticulus?
Fructifilus, the Phyllocoptes fructifilus.
Yeah, yeah, that's it.
Fructifilus, thank you. Yeah, Phyllocoptes fructifilus, they definitely are population diversity in the way that they get isolated in some areas and in the way that are feeding. Again, it's a mite that is only 100 microns. And very simple in an amount of genes. There is a guy, Pavel Klimov, in the University of Purdue, working right now with genomas. And I hope that he will run the genoma of Phyllocoptes fructifilus. Maybe somebody is already working on that. That will help us also to see how much variability is inside the family. But I'm sure, in base of what we are learning from Asteria toshikella and all their virus associations, that we can use that information in our bad guy.
Mm-hmm.
Oh, there is one thing also, Dave. And this is another thing that some of you in the field may can look. There is a possibility that these Phyllocoptes fructifilus may be using also aphids for transportations. When the aphids are feeding on the flowers, the mites maybe are grabbing the legs of the aphids to go to another places. That is another possibility too, in base of now this pattern, because now we have a pattern of behavior. Now we can guess, oh, this mite is capable to do this too.
Oh, that's kind of scary. So we have to control aphids too.
Yeah.
Oh my goodness. Oh, here comes Mark. Hold on.
Yeah.
What you just said is a bombshell. You know, we had found that in pans that are certain colors, that Eriophyid mites seem to land in those pans more than others. But the colors that they seem to land in are the colors that attract aphids. Well, we just ignored the aphids in the pan. So you're telling me that the reason why there may be more mites in those pans is because of the damn aphid?
Yep. Yeah, it could be a very strong possibility. We have
a discussion
in
and a
others.
thesis is about to change. Okay?
Yeah.
We'll be in contact.
Okay,
Okay.
thank you. Filter some aphids to see how many Eriophytes you find.
Oh, Ron.
No, this is fantastic. I need to say I love it. I really love it. But one thing that I talk with John and Ramon and with all of you guys, think about this. Eriophyte is just a bus. The virus is the driver. And the bus, the virus already know what to do with the driver. If we can attack that bus that not go that road, perhaps we will hurt the virus.
So since the virus drives the bus,
yeah,
resistance could be conditioned in the virus by affecting the mite. Interesting concept.
Yeah, there is another thing that we know in other family. There is a family called in the family Tenupalpidae, the genus Brevipalpus, and Brevipalpus is associated with leprosis virus on citrus. One of them is cytoplasmatic virus. We discovered that the cytoplasmatic virus not only made the life of the mite shorter, but made the mite to put more eggs, from 25 eggs to 50 eggs. And perhaps something of this also is running with this virus and Phyllocoптers, when the Phyllocoптers that do not have the virus have a longer life, less progeny. When the one that have the virus maybe live less and put more eggs. That is perfect for the virus. That is a possibility too.
Oh, here comes Francisco with a question.
Okay.
Hola.
Oh, okay, Francisco.
My question, actually, I'm a bit concerned with, are these hypotheses, or do you have some data to support?
No,
this?
All these that we are talking are data in other Eriophytes, in other Erinions and other things. And what I am doing is putting all the information that I know of similar Erinion, trichome, virus, mite, put it all together and say, okay, maybe there is a pattern here. And that is the reason that I did show you the photos of the trichomes. So you saw the photos of the trichomes of the 4 plants, rose, beech, oleander, and all that are the same. And by the way, guys, I have 40 more plants that are doing the same.
Ron, would be very nice if you can put all of this in a review. So because we are envisioning the future. So
yes,
depending on these hypotheses and facts, then we may drive research. So it's very important to distinguish between these facts.
Yes.
That's just a recommendation.
Yeah, Francisco, you have the ability that in the United States, there are 3 good research on virus Eriophyid mites. One is the wheat situation. The other is now with the lychee situation that not involve a virus, but is giving information, and the rose.
Yeah. Thank you.
You're welcome.
Wonderful. Thank you very much, Ron. We need to move on to our next speaker, but it was very entertaining and elucidating, if nothing else.
Yeah.
Kind of scary, but that's okay.
No, but we are fine, and I am here to help everybody about the what I can about the Eriophytes. And good luck, guys, because you are doing an excellent job, and I think this is the way to attack. Thank you.
Thank you very much.